One of the hallmarks of life is specificity, that molecule X interacts with molecule Y and not with molecule Z. What we're going to do in this video is unpack that for the specific example of the steroid receptors. It's a cool story, and I look forward to telling it. So in a previous video, I talked about this different sorts of molecular forces that determine how biomolecules interact. And I broke non-covalent interactions into two broad categories: polar interactions and non-polar interactions. And the big difference between these is this polarity, right? Let's say the non-polar first. Non-polar interactions, as long as a hydrophobic thing is next to a hydrophobic thing, they're going to stick together. There's no sort of specificity to the direction. Whereas with polar interactions, you have specificity. You have some polarity, you might say, to the interaction. So a positive charge can interact with a negative charge. That's great. That's awesome. That's favorable. But a negative charge is going to repel a negative charge. So it's not a willy-nilly anything can stick to anything in polar contact world, whereas in the non-polar context, basically anything can stick to anything. This means that these two sorts of interactions play different biological roles. Non-polar contacts, non-polar interactions tend to just make molecules stick together in a very generic way, whereas polar contacts really give specificity, right? They say you can only stick if you meet certain criteria. And we're going to see that example in spades here with the steroid receptors. So let's introduce our molecules and our biology here. So many animals exhibit sexual dimorphism. This is a peacock and a peahen. They are the same species, yet they look pretty different because of this sexual dimorphism. And at a biological level, much of this difference is actually driven by the level of the hormones testosterone and estradiol. So estradiol is higher in the peahen, testosterone is higher in the peacock. And the way that these steroids, these hormones work is through receptors. So if estradiol comes into a cell and a cell has an estrogen receptor, that it can bind, the estrogen receptor can bind to the estradiol and then translocate to the nucleus, where it will upregulate genes, say brown feather genes, corresponding to the peahen phenotype. Whereas the testosterone interacts with an androgen receptor, and the androgen receptor recognizes different genes and will say turn on green feather genes in a peacock. Now, before we dig into the very cool and simple, honestly, biochemistry of this, I want to talk about a not simple thing, which is the relationship between, say, testosterone level and social constructs like gender and the like. It's extraordinarily murky. There's no simple rule, even though there are some very common myths in society about how testosterone makes manly men and stuff. I just want to make a plug for this book, Testosterone Rex by Cordelia Fine, which is just this amazing exploration of this topic, sort of intersection of sociology and of biology and social constructs like gender. Great book, extraordinarily funny. I totally recommend you check it out. Okay, so let's talk about these at a sort of molecular level. So these two hormones, estradiol and testosterone, are extraordinarily similar to each other. This is estradiol here. It is derived from cholesterol, and it has basically a big hydrophobic bricky thing. Whoops, we're going ahead. Wrong way. Okay. Big hydrophobic brick thing, and then it's got two hydroxyls hanging out the side forming hydrogen bonds. And so they can potentially form hydrogen bonds there on those sides. Testosterone looks pretty darn similar, right? It has the similar architecture of a giant brick with polar stuff hanging off the sides. But you'll notice the difference really does come down to this region, which is called the A ring, incidentally. And in this A ring region, instead of having an aromatic A ring with a hydroxyl like estradiol, testosterone has a non-aromatic A ring with a ketone hanging off of it, or a carbonyl. And so they're pretty similar looking. There are some geometric differences down here, but overall, if you were to say, oh yeah, do these bind to the same site or different sites? My first guess would be that they would bind, that they have similar enough geometries and similar enough ability to form contacts. I'd be like, yeah, these can probably stick into the same site. But receptors can, in fact, tell the difference. The estrogen receptor responds almost exclusively to estradiol. The androgen receptor responds almost exclusively to testosterone. That is a remarkable and cool fact. This has a name, or the biological concept here is that of specificity, and it's when a biomolecule selectively interacts with its target rather than alternative target molecules. Now, if we look at the receptors that are doing the recognizing, they're quite similar to each other. I've overlaid the two of them. The androgen receptor is in gray, the estrogen receptor is in blue. They are the result of an evolutionary duplication. So there was an ancestral gene that duplicated in the genome, and then after that duplication happened, both the androgen receptor and the estrogen receptor sort of specialized on which steroids they could recognize. But both of these receptors in their architecture have a bunch of alpha helices and then a deep hydrophobic pocket inside where the steroid can bind. And when it binds, it then triggers actually a conformational change in this region that activates the receptor. So I'm actually going to focus exclusively on estrogen, the estrogen receptor from here on out. I could tell an almost identical story for the androgen receptor, but then this video would be even longer than it's going to be. So the story we're going to tell for the estrogen receptor could translate just as well to the androgen receptor. Okay, so what we're going to ask is, how does the estrogen receptor recognize estradiol? So I'm showing the receptor again. I'm now showing the pocket and some balls and spheres. What we're going to do is we're going to balls, spheres and lines and sticks, we're going to zoom in on that pocket. So here we are zooming in. And now we slice away the protein, and now we can look at this and say what sorts of interactions are formed. And this is a good chance, if you're working on your biochemistry type skills, for you to actually pause the video and ask what sorts of interactions are being formed between estradiol in light blue and the pocket, the receptor in dark blue. So here's a chance you can pause. And I'm assuming you now unpaused. And so let's talk about the interactions. So one set of interactions is up here, and these are the hydrophobic or non-polar interactions. And this is basically a bunch of carbons and a bunch of non-polar hydrogens that are all stacking together, excluding water, having a nice hydrophobic interaction surface. Over here, we have the sort of right side of estradiol is interacting. It is donating a proton, to an H bond. And then on this side, we have that aromatic A ring that's both donating and accepting. The hydroxyl is accepting a proton from an arginine, and the hydroxyl is donating its proton to a glutamic acid or glutamate. Okay. So those are the interactions that are being formed. Now let's actually do this in sort of a counting way. Can we sort of start building a table? So when estradiol is bound, what H bonds is it forming and what hydrophobic surface? Turns out it's forming hydrogen bonds to the protein, and its hydrophobic surface is baried. And then if we ask when the estradiol is bound, what's happening with the protein, we would say it is forming its hydrogen bonds to estradiol, and it also has baried hydrophobic surface, right? So we're counting, those are the sorts of two things that are happening. Now, we're talking about a binding reaction here, right? This is a non-covalent thing that can form and unform. So to do this analysis, we actually really have to compare the bound and the unbound state, right? We have to say what's different between these two states if we're going to say what's causing it to bind. So what I'm going to do is I'm going to take the estrogen receptor and pop the steroid out. So now I've got these two next to each other. And we're going to start thinking about the nature of the interactions in these two states. So when these are unbound, what's going on? So maybe most obviously, the hydrophobic surface is exposed, right? We've taken the steroid, we've popped it out, we've stuck it into water. Now it's exposed. We pulled the steroid out of the binding pocket. So now the binding pocket's exposed. So we've clearly changed that. What about the hydrogen bonds? To think about this, let's add something else to our picture, and that is water. Remember, water is 55 molar, so there's just water everywhere. And so when we're thinking about, well, what hydrogen bonds are being formed in the unbound state, we have to worry about what the waters are doing. Actually, sorry, I should have said that. Sort of emphasize what I just did. So what I just did is I sliced back into, I'm going to go back actually so we can do it. So here's the protein in the bound form wrapped in water. We're going to slice down inside so now we can see what it's interacting with. And what hopefully you can see in this pocket is there's a steroid bound in the pocket, and then we got some water lurking outside here. But overall, the protein's interacting with the ligand, with the steroid's interacting with the protein, and the water is kind of lurking on the outside. If we look at the same picture for the unbound form, we now see there's a bunch of water in this binding pocket, and that the hormone is surrounded by water. And further, if we're really doing all of our accounting correctly, we also need to account for the fact over here that waters got displaced out when that one bound. So now we've got more water hydrogen bonds forming on the left than on the right. Okay, complicated. So let's now put this back into this accounting table. When estradiol is unbound, what hydrogen bonds is it forming? It's forming hydrogen bonds to water, right? And when the protein is not bound to estradiol, it's forming its hydrogen bonds to water. Let's show this schematically. So what I'm doing here is now, instead of drawing all the structural details, I'm drawing a very simple version of this binding pocket. The black edge here, this is the pocket itself. And then we've got our steroid bound in the middle, our hormone bound in the middle. When it's in place, it's forming a hydrogen bond with this histidine at the back of the pocket, and it's forming hydrogen bonds with the arginine and glutamate at the front of the pocket. And then there's waters that are floating out here. When the steroid comes out of the pocket, when the hormone pops out, well, we've got water that comes in and makes these hydrogen bonds, and we've got water that makes this hydrogen bond. And we've got water that makes all of these hydrogen bonds. And what this shows basically is when estradiol binds, hydrogen bonds are swapped. They're not gained or lost, right? You're swapping hydrogen bonds between water and between the pocket and water. There's a swapping happening, not a gain or loss. This contrasts quite strongly with the hydrophobic effect, right? The hydrophobic effect is totally in effect here. All kinds of hydrophobic surface area was just buried. That's really favorable. Whereas over here, the hydrophobic effect is not operating, right? Water's everywhere, so the hydrophobic effect is not favorable on that side. So we can write it this way. We can say that when estradiol binds, the hydrophobic effect favors binding, right? It's great to bury all that hydrophobic surface area. But the hydrogen bonds, to a first approximation, actually don't have an effect at all, right? They're being swapped. You're basically swapping water hydrogen bonds for protein ligand hydrogen bonds or protein hormone hydrogen bonds. So there's a swapping, but no real strong effect on binding. So why have all these hydrogen bonds? Like if you look across molecules, there's a bajillion of them, and they clearly are important in some way for the biochemistry of these interactions. Why are they there? Well, to probe this, let's actually look at testosterone binding. So here I'm showing estradiol in the pocket, and now what I'm going to do is jam testosterone in. So this is not a real structure. This is just me aligning a testosterone and sticking it into the structure. And I just did it. I'm going to pull it back. Did it. Going back. So what do we notice about the testosterone binding versus the estradiol binding? And what I notice is a big difference right here, right? This, because of the geometry of this, it's not able to form the hydrogen bonds that estradiol does to that aspartate and arginine. Sorry, glutamate and I can do this. Glutamate and arginine. It lost hydrogen bonds. That's the key point. And so let's do one of these diagrams we did for estradiol binding. So over here, like estradiol, the hydrophobic effect favors binding, right? There's great hydrophobic interactions when it's bound. Those are there for both estradiol and for testosterone. But when testosterone binds, it actually loses hydrogen bonds, right? It went from being unbound and having all kinds of fun hydrogen bonds with water. This had nice hydrogen bonds with water. Those are now lost. So remember, for estradiol, hydrogen bonds basically swapped. We swapped water for protein hydrogen bonds for estradiol. Now we're swapping water for no hydrogen bonds. So that means that losing hydrogen bonds is disfavoring binding. So we can put it into this table, right? We said that with estradiol that the hydrophobic effect favors binding. The hydrogen bonds have no effect on binding. In this table, now we can say that testosterone, the hydrophobic effect, continues to favor binding. So you're still bearing greasy surface area, but hydrogen bonds now disfavor binding, right? You'd rather form hydrogen bonds back away unbound with water than losing those hydrogen bonds, jamming it together. And so this gives us this rule of thumb in biomolecular specificity that the hydrophobic effect tends to make molecules stick together, whereas hydrogen bonds don't really cause things to stick together. And it's because hydrogen bonds are so easily formed with water. You're basically swapping water to binding or whatever, but there's always hydrogen bonds can be formed with water or with protein. They're not really the thing that tends to be driving sticking. But unsatisfied hydrogen bonds are key because they prevent the wrong things from sticking. Because the testosterone has the wrong hydrogen bonding partner type thing at the A ring, it can't stick to this receptor, giving it specificity. And what this ultimately means then is very small molecular differences. So in this case, this tiny little difference right here can have profound effects to the level of the biology of these organisms. And a lot of what we're going to be doing in the next video and the videos thereafter is trying to take apart and understand those. Okay, that's what I've got. Hopefully this gives you a better notion of where biological specificity comes from. Again, we're going to be unpacking this in future videos, and thanks for watching.